Okay, so today we are very pleased to welcome Sheng-Yang He from Duke University from US in North Carolina. And so Sheng-Yang is here this week or this 2 or 3 days in the context of the Tulip Graduate School, as you may probably know. So the master of this functional biology and ecology master is organized each year 2 weeks, one in June, the other in September, and we have some guests that comes to propose some courses and to also some roundtable journal club with our student, and we also asked to the guests to give a seminar, and we are really pleased to welcome Sheng-Yang today for the master and for this seminar. So a few words about Sheng-Yang. So you did your PhD in Cornell University. Then you moved to Michigan State University, and since 5 or 4 years now, in 2020,
the middle of
2020. Okay, so you are now still group leader for a long time in the Duke University, and you work still on plant pathology, and you work on that. You are certainly one of the leaders in this field. And interestingly, since this last year, you were interested by the effect of environmental changes on immunity and how that can be controlled, maybe microbial interaction and plant immunity at the plant level. Okay, so thanks for coming and for the master and for us. So please.
Well, thank you very much, everybody. So it's really great to be here again. For those who don't know, I was here before, long time ago, before many of you actually arrived here. So great to see some old colleagues here, but also new one. Yesterday I had a very good discussion with many colleagues here. Learned a lot. Jean-Philippe asked me to deliver a seminar. My lab actually now doing quite different things. Pathogenesis, microbiome, and then some work on climate effect on the interaction. So I'm going to focus on two short stories today, just to give you an example what we're approaching this right now, probably in the near future as well. So that's my title. And this doesn't work, but the pointer works. So I'm going to advance this one here. I don't have control here. Sounds like the new technology is always there. Okay. Oh, here you mean? Touch here? Oh, good. All right. So for those who are not very familiar with the field of plant-microbe interaction, we actually have a lot of progress in the last 4 decades now in terms of molecular dissection. Try to understand how pathogens cause disease and how plant fight back and to be resistant. So it's a continuing battle, and we are dedicated to understand in different crops and model species. So a lot of work is being done in bacterial pathogens, which was really the early on work. Many of this is done here. Really good to see Stefan here also and Laurent here. A virus, that's initial work, but then right now there's a lot of work on fungal pathogens and oomycetes and nematode insects even, and parasitic plants. They all look quite different. You know, we think like the parasitic plants and insect quite morphologically at least different from microbial pathogens, but what we learned the last few decades, I can say this, there's a common theme in all these interactions, which actually go beyond plant pathogen interactions. So if you look at human pathogen interactions, some of the basic principle we learn and other people learn converging to this particular picture here. This is a plant cell or animal cell. You have these microbial pathogens, the insects, maybe parasitic plants. What they commonly do is basically send a bunch of molecules into the cell. And many of them actually live outside the cell. So they're extracellular microbes. Essentially, they don't come in. Pretty dangerous place here. But they are sending these so-called effectors. So these are proteins, DNA, and mRNA, and secondary metabolite, so all kind of different mix to essentially subvert whatever is resisting that microbe. Right? So that's the take-home message here. Well, we know this is an important process because the delivery system Each type probably deliver different ways. So insects, as you know, they use stylet to inject the molecules into the cell. So it's nematode, but the bacteria has specific types of secretion system, a lot of them, or type 2, although they don't inject. We don't know how this is delivered into the plant cell, but if we knock out genetically genes that are delivering these proteins, you will have a dramatic effect. So type C secretion, you can actually see the needle-like structure. This is built by bacteria involving quite a few proteins. You can see under the microscope, but if you knock out this pillar-like structure, the resulting bacteria will be non-pathogenic. So we know essentially whatever delivered into the plant cell collectively must be very important, right? So that was early on kind of realization, the importance of the delivery system. Because of that, I think what we community collectively thought, if these are actually great probes into the molecular basis of disease susceptibility, it's the entry way to understand why pathogen could cause disease in plants. We can understand what each of these factors does in the cell. And later on, we found that these molecules could be DNA or RNA or proteins mostly. They're actually very interesting probes into finding regulators of many aspects of host biology. So it could be plant biology, it could be animal biology, because they find, transcription factors, kinases, and all stuff that are actually mostly involved in immune responses. So this is a really good way of finding missing links in the plant or animal immune system. Of course, what we really want to do ultimately is to identify these disease targets or susceptibility targets that could lead to disease control measure. We don't do this very well. We write this in the grant proposal, but it's a selling point. But I will give you a couple of examples today that this may be upcoming as a final goal. Okay, so we have been working on this very simple system involving a bacterial pathogen called Pseudomonas syringae, and this is infecting tomato in the field. The strain we work with is called DC3000, which was isolated by Diane couple initially in Canada, but Brian Stassek's lab made it into a Arabidopsis pathogen in their efforts early on to clone so-called disease-resistant genes. Okay, what we decided to do is not to be involved in ET or affected to immunity or disease resistance, is to actually study the other side of the interaction, which is in genetically susceptible plants, how pathogens are able to cause disease. So we have worked on this system quite a bit. This bacteria actually mainly infected the above ground parts of the tissue, mainly the leaf, fruits, and stem. They can live on the surface of the plants, including leaves, but in order to cause disease, they have to go inside the leaf, and this is called apoplast. So these are in between these mesophyll cells, and they live as an extracellular pathogen. Okay, they don't invade the cell, and it's quite common for plant pathogens. So we have studied, different stages of infection. For instance, we spent quite a bit of time trying to understand how bacteria get into the plant interior and how plant actually gut cells have defense mechanism to keep the bacteria out as a first line defense. So that was an immune function of the stomata, which normally you think about for photosynthesis, but actually has multi-faceted role in plant biology. We spent a lot of time to understand how these effectors manipulate plant cell physiology. This particular pathogen actually secrete, I would come to that, about 30 or so effectors, so a lot of them. And so that was quite challenging, but we have made good progress as a community. In this particular system, we can come to a conceptual model come up soon. And then I also was fascinated by this toxin produced by DC3000 and related strains in tomato cultivar. And that this turns out this toxin, if you look at the structure, it's actually molecular mimic of jasmonate, specifically J-isoleucine, which is the active form of jasmonate. So this is a case where a pathogen hijack the jasmonate signaling system, does a lot of things, but one of the things it does is to counter the stomata closure that plant has evolved to recognize these bacterial molecules to close it, but this is to prevent it so that pathogen can invade and then proliferate in the substomatic cavity. So we have done quite a bit of that, and I think one thing that's limitation, quite a bit significant limitation, is we have to use this system, grow plants in this very static condition, obviously, just to have comparable results across the lab. And I think most labs across the world use this kind of system. For adults, we use 21, 23 degree because Pseudomonas prefer 23 instead of 21. But 60% relative humidity. And most of the data we publish is under this condition. So this only reflects the fraction of biology, right? So if you look at outside of the plants growing, of course, each plant is experiencing much more each day. Temperature, humidity fluctuate and other things. And so that's what we decided to take a new direction about 10 years ago to try to see if we would change some of the abiotic conditions that deviate from our traditional way of doing, does the interaction still hold or does the model still hold? Okay. And if it's not, where we can improve and things like that. But this is getting to what we really, about what's going on in the outside. The reason is we all know from past literature, these individual abiotic conditions can have dramatic effect on pathogen itself in vitro, right? So that's what a lot of disease forecasting model was based on, the in vitro effect of abiotic conditions. Of course, plants respond to these abiotic conditions in very dramatic ways, but also combined, have more combined effect. And so that's even mystery to a lot of people. This is a slide I put just recently. I grew up in China, so a place like pretty hot and humid. It's like Toulouse, I guess, in the last few days. But I know this concept, the climate condition on disease very, very well, because I grew up in a very small village. And one side I remember was rice field, and the other side was cotton field. Every summer I would smell these things, and these are sprays, I think, whatever they spray to control plant hopper, I think, and rice blast. So rice blast disease was very familiar concept to me when I was a kid. But I know them from, we were not farmers, but our neighbor were farmers. And then we knew that even though the same cultivar they plant and same presumably pathogen in the field, but they don't get disease every year, right? So only get disease certain year because they have this forecasting, the rain and the temperature. And so the effect of the climate on disease is very obvious for even, lay people. This is a few slides put together. I'm traveling around the world quite often these days. In China, this particular disease, Fusarium hepatic, which is really severe disease of wheat, they have consecutive epidemic in the last decades. Every year I can count on this. One of the reasons is because of the practice. They don't remove they don't till anymore. And they don't want to burn the straw anymore. So there's a lot of accumulation in inoculant from winter. This is warm now. So in spring, it's really heavy primary inoculant. When the weather is really nice, it just destroys the wheat field. Major problem. Also, there are reports, obviously, pathogens evolving much faster than plants in general. The rust fungus, for instance, have these thermal-tolerant variety already there. And so plants have to catch up, how to deal with that. This is also a very strange phenomenon they cannot explain with the Curling model. So Pseudomonas infection usually is favored by high humidity after rain, so that's very common knowledge, but also cool weather. So if the temperature usually go above certain degree, you shouldn't get any disease, okay? This was a slide sent by a colleague of mine, Yu-Fu Zhao from Washington State last year, I think. This is in the middle of July with temperature 100 degrees Fahrenheit, which is really high. And that's where the outbreak happened in the cherry bacterial canker caused by Pseudomonas syringae, and nobody anticipated this right now. So I think this is like emerging phenotype where the climate is really go beyond the prediction. And I think that has some unpredicted consequence or probably a pathogen on the plants or the interaction between them. So this is a complete knowledge gap. You probably have seen this report from FAO a few years ago now, predicting major diseases could become more severe. We don't know whether it's going to be true or not, but that's based on the pathogen biology that this can to be quite severe. I would mention that a lot of invasive pests now are moving northward. Of course, you know that Europe is the same way. So I heard that the grape has to move northward here in Europe. So France is worried about this, I guess. No? Okay. Anyway, so I want to give you two examples to just illustrate how we are getting into this area, try to understand how different abiotic conditions going to affect plant pathogen interactions. There's many abiotic conditions you can see on my second slide. So I'm going to talk about two. One is high humidity, and the other one is temperature that's above normal. Okay. So first, high humidity, and this work was spearheaded by former postdocs, Shufang Sheng, Kenya Nomura. They have since left my lab right now. And this was done in the context of, not on purpose, actually, initially, was to understand how these effectors individually manipulate the host pathways. So that was the goal, okay? And so there are 30 or so effectors in this system. So we and others have been take each of these effectors and do all kinds of molecular work, try to identify the targets, you know, blah, 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 okay? And what we have done 15 years or so, I think, collectively realized that initially we thought these 30 effectors can attack like 30 different pathways or maybe 15, or like many different things. But in the end, we think mainly they target two pathways in plants, okay? One is immunity. So many of these effectors, we call them immune suppressing effectors. That means they tune down the immune responses of the plants. That makes sense? To infect the plants, you have to kind of suppress that level. And that happens in human pathogenesis. That a lot of Salmonella, Yersinia, they tune down our immune system as well. So this is very understandable. This concept now, even though it started with bacterial pathogens, it's very common for other pathogens, even insect and fungal pathogens. So I think that's a universal concept, even though each pathogen probably does it differently. They attack different components of the plant immune system. One of the things they want to do is tune this down. The other one, what we recently described, is a process where some effectors, we call them water-soaking effectors, they actually cause water accumulation inside the leaf. So if you look at the water leaf, it looks like water soaked. Okay, and this is very common actually in not only bacterial pathogen but other type of infection, including fungal pathogen or mycetes, etc. So I'm going to talk about this today because this is affected by humidity. This action of water-soaking effectors will be most obvious under really high humidity, like simulating after rain kind of real situation. Okay, so what is water soaking? For those who don't know about water soaking, I actually made the slides. It took a while for me to make this one, so it's this is a very dramatic way of saying it. Basically, normally this space is filled with air, right? You know, this is important for photosynthesis. If you have water there, the CO2 diffusion will be quite limited, and so plants don't like that at all. So there must be a very precisely controlled mechanism to keep this space air filled. I think that we don't understand precisely how that work. And yesterday I was talking to the Orange group. It was interesting, you know, like hydrosol may be also involved in the making sure that space is well regulated, so it's not congested with water. So I think there's probably multiple level of regulation. But microbes probably don't like that because they need the water. I mean, so that's what we think. And so that's what happens is this particular bacteria is creating this. But this is only most effective under high humidity. I'll come back to this, why, okay? And so it doesn't look like this. This is a normal leaf, and this is basically water-soaked leaf. And this is usually early stage of infection. What happens is that this washout area usually come later with necrosis. So when the disease progress. A common phenomenon in plant disease. And in the case of bacteria, so different pathogen may have different, effectors to do this. And there may be many ways of creating that. But in the case of bacterial pathogen that I'm studying, there's a particular family of effectors. We call them AVIE or DSP. Names are not important. Just these effectors are there. These are very conserved, actually, in many strains of Pseudomonas syringae, but also beyond Pseudomonas syringae. We think they're probably probably most important variance factors, actually. Okay, if you genetically knock them out, in the case of Ovinia malabra, which cause apple and pear fiber blight, which is a severe disease sometimes. I don't know in Europe it's a disease or not, but in New York and Michigan, now it's really pretty bad. In China, especially on pear. Pear is super susceptible to this, and they're really worried about this. But if you knock out DSPE, this particular effector, you basically kill the disease. There will be no disease. So the single effector killed it, so it's pretty dramatic. Most effector deletion don't show much. Stefan probably can confirm that it's very frustrating that for us, if you knock out individual effector genes, you usually don't see an effect. So there's a redundancy going on. But for this one, really dramatic. AVIE in certain strains of Pseudomonas syringae, for instance, kiwi bacterial canker, which is also quite severe in Europe now. You see quite dramatic effect also. So these are really very crucial variance factors. However, for a long time, very challenging to study, partly because the proteins are really big. It's 200 kilodalton. I think it's probably largest effector protein ever in plant or animal pathogen included. So it's very hard to do any experiment, cloning, yeast to hybrid, and all kinds of stuff. Very toxic also. If you put in any eukaryotic cell, it kills them. A lot of techniques available for effective biology studies are not available for this. So this was the project I wanted to do when I became faculty member, which is a long time ago. So on and off, I gave up and I don't give up. And so it's like faculty member probably know what that means. Like you know this is the important thing to work on, but you can't work on. And so but we persisted, but no major breakthrough until very recently. Essentially, we were able to express AVIE in the plant cell now. And you can see that actually AVIE GLP fusion protein, so that it can see the fluorescence of this protein to track them in the plasma membrane of the plant cell. But in a punctate structure, it's not a uniform. So we don't understand why it's punctate, and we still don't know what that means. And about 3-4 years ago now, there was the AlphaFold came along, which is the AI, which is super cool. And we did not give up. We said, okay, we go through all the effector sequence, maybe pump through this program to see what new things come up. So AVIE and DSP came back to be very striking results because they predict to be a channel like proteins. We couldn't believe it, and then we feed it was I think 50 or so members, like there's different members of this family. They all come back with this kind of prediction, so it's pretty confident that there's something's going on here. This is side view here. You can see this is N terminus, this is C terminus, the beta barrel structure, and this is the top view. You can see a pool. It's just a prediction right now. We did a cryo-EM also to verify that you can actually see a pool in the middle of the protein from the top view. We're still working on to resolve the whole protein. It's quite difficult to the resolution we want. Anyway, so that gave us a new hypothesis. Maybe these proteins form channels, which we never imagined before. So we did a bunch of electrophysiological and liposome and protoplast and other implant experiments. I don't go, these are published, so I don't want to go through all of them, but just to show a few results. But they collectively consistent with the idea that these are actually channels in the plasma membrane of the plant cell, and whatever they're doing is to accumulate water and solutes. And so example of electrophysiology, so this is what you do is you inject oocyte with a cRNA of either water control or a mutant of DSP, for instance, or the wild-type DSP. If DSP encodes a channel that targets to the membrane, it will go to the membrane and allow the ion to go through. Depending on what ion goes through, you can measure the voltage change or membrane potential change. And in this case, it actually works really well. You can see DSP causes nice ion conductance across the membrane of the oocyte. And in these experiments, though, we observed a very strange phenomenon. So if you look closely, this is a control water-injected oocyte, so they look smooth and they're fine, okay, round. In the DSP CNA injected the oocytes, you see these things here. These are the injection site where the shell, the outside shell is loose, it is weak, okay? What you're seeing is the membrane actually kind of pushed out from the inside the outside. This usually means, if you have trained eye, means the internal water pressure is higher than outside. And when that happens, usually the osmotic pressure outside is lower than I may be reversed. Anyway, there's osmotic pressure imbalance, so that the water goes in there and then it tries to increase the cell volume. Because this is a weak point, so it pushed out to the membrane. So that was exciting because there was a classical water channel phenotype, essentially. If you put up animal or plant water channel into oocyte, that's what you see, okay? So to test that directly, we basically did an experiment to artificially lower the median osmotic pressure to very low, to 40 milliosmol, compared to the initial 200, which is the normal electrophysiology experiment, to see whether now we can see actually bursting of the oocyte. Because if the oocyte cannot hold the volume increase, it would just burst. So you can measure that. In fact, it happened within 10 minutes, all oocytes burst. If we infiltrate with inject with the Y-type DSP cRNA, the DSP KK is a virulence mutant. That it does not do virulence. We want to see whether it's connected to the virulence activity. It kills it, okay? And then the water, of course, has no expansion. And this is a video. I don't know if I can play this or not. Let's see. So this is the control within 10 minutes. And this is the DSP cRNA injected. They look like a balloon, they just pop out. So it's pretty dramatic phenotype. Anyway, you can also do this with protoplast, Rhabdopsis, which express AVIE and an inducible promoter that is not constitutive. You can have transgenic plants. You isolate protoplast, you put into a hypoosmotic solution. Basically, they increase protoplast will increase the size when you have expressing AVIE, and it just burst. So the protoplast is very easy to burst compared to oocyte. So the volume increase is not dramatic, but they burst. So anyway, we think these are channel proteins. So once we identify that, we thought, this is a chance to come up with a solution to inactivate this channel, because you can develop inhibitors that could inhibit these ion channels. So we screened for chemicals that could block this channel. And I'm not going into this, is actually very labor intensive. We started with oocyte-based screening and then did implantar infection. And we were lucky to come up with a particular group of compound called PAMONs, or polyamidoamine dendromer. These are synthetic polymers. You can actually program it to different sizes. So certain sizes of PAMON derivative can actually indeed inhibit the channel activity in the oocyte. So we now test the implant activity here. So this is actually infection experiment with Ovinium lavra that cause fibroid disease, as I indicated before. This is a immature pair assay, so pair is really susceptible, as I said before. This is a wild-type infection. It caused a lot of necrosis of the pair. This is a DSP single mutant in bacteria. As I told you, it actually knocked out the infectivity, so it's pretty dramatic effect. But now if you combine the Y type and G1, which is one of the one that actually fit the pool in the computer, actually completely block the activity of the infection. So this is really suggesting the model that this could be potentially a channel. We also did the infection experiment against the kiwi bacteria canker. And you can see here Y-type infection in the leaf look like pretty bad in this case. If you combine with G1, the chemical, you basically eliminate symptom, but also you see 100-fold, this is log scale, so 100-fold bacterial population drop. So this is a very dramatic effect. This has been taken to a field trial right now. And this is a colleague in China helping us to see whether it actually work in the field. So that's unpredictable. So, a lot of times you have very beautiful results in the lab, and the look at the disease is very dramatic. These are big trees, and these are bacteria coming out from the trunk. So I don't know how to deliver this compound, but they're trying right now using different approaches. So you may ask, what has something to do with high humidity, right? So this is a step where bacteria actively try to create water-king. But why do you need high humidity? Do you have a guess? So the bacteria is actually making water here. Why do you need high humidity? Okay, time's up. What we think is because, if the air humidity is low, during the day, the stomata are open anyway, so it will evaporate. I think we can actually document that. Within 10 minutes of lowering humidity from usually 90, 95 or so to 60%, the water created here like this would disappear completely. And leaf look like perfect. It's not like dead or anything. So it's just like water going in and out. So I think that's one of the reasons why the high humidity is needed, but also, there are other mechanisms that you can close the stomata, and so I don't want to go into all these complications. But there's a lot of detail we need to figure that out. Okay, so that's part of the high humidity. So this is a case where you need a high humidity and then a passaging mechanism to maintain a process that is really crucial for disease outbreak, right? So that's called disease triangle, I guess. You need the host, you need the pathogen, you also need the environment condition to work together to make sure that the pathogen evolution work. All right, so we have finally discovered a core function of this mysterious family of effectors. And we have identified some inhibitors that could be used, hopefully in the field in the near future. These are three people did the experiments. Kenya Nomura was in my lab until recently. Philippi Andreza was in Ke Dong's lab, and then Jennifer Chang was in a structural biology lab. They are at Duke. I want to say a few things about No King. AVIE was discovered in 95 by No King. I don't know how many people know No King. He was a giant in plant pathology, molecular plant pathology, passed away many years ago. He cloned AVIE as a gene that could elicit the hypersensitive response. There is a hallmark also called effector immunity or disease resistance. And I was supposed to join his lab for postdoc. I didn't go there eventually for some other reason. So it was a really nice personal satisfaction about this because I think he would like this result. Okay, so I talked about humidity. Obviously, in nature, plants experiencing other abiotic stresses. So we know how projects are working on temperature, salinity, and nutrients, and also pH. This is a growing area in my lab right now. I try to see whether any of them have an effect on disease progression. So the four questions, we're pursuing. So we have to know whether these other abiotic conditions actually affect disease susceptibility. So if you read literature, it's not really clear. And the condition people use also, anyway, we need more work to kind of refine these conditions. And if it does, then, you want to know whether it's affected plant or pathogen or the combination of the two. But for us, the questions 3 and 4 are most important. If we identify the plants are impacted, we want to identify which molecules are being affected. So, okay, so that's important goal. And hopefully we can repair that whatever sites or impact to make plant resilient to that particular abiotic condition. So these are the 4 kind of workflow. We're at very early stage on most of these projects. We went through temperature a little bit further, so I'm going to give this an example of how we do it and get your input on that. So temperature effect on temperature. Wow. Okay. This is supposed to you all know this, so I'm going to skip this. Increasing, especially in France right now, today, I guess. All right. So in the plant immune system is well studied. A lot of people dedicated to this in the last 4 decades or so. We learned a lot. So this is very a simple summary of what the immune system look like. We have so-called a pattern trigger immunity, where plants recognize conserved pattern like bacteriophyllum, which many bacteria have this, or chitin with fungus and all mice have. So these are very basal level kind of recognition. Usually we call basal immunity. Most plants have that. And then pathogen comes in, usually overcome this layer of immunity, which I kind of summarized before. And this pathogen have to do this to be able to be infectious, okay? And then plants defend against that level pathogen attack by developing so-called effector immunity. These are a branch of immunity only target to pathogen because it's triggered by effectors, which is only present in pathogens, not in microbiota, for instance, okay? So, and these 2 branches link to these defense hormones, salicylic acid, jasminic acid, ethylene, or ABA. You know, this is a very complicated interplay. I don't think we learned this is a very simple view. I think in 10 years we have to revise this thing, but it's a network of things, and different plants probably operate different ways, and even in the same system probably operate at different conditions. Okay, so effect on the immune system actually is well known. I mean, if you go back decades ago, you know, with tobacco mosaic virus in tobacco was shown the immune receptors that recognize TME is susceptible to temperature. So it's a classical example of this called ETI, or effector immunity, is very susceptible to temperature. There come very interesting data, molecular understanding. You know, the initial, this I remember really well, Jiang Hua at Cornell University. I don't want to go into this. They did a genetic, beautiful dissection and pointing to these NBLR, these are resistant proteins, or NLR, involved in the effector immunity, as themselves are temperature vulnerable. And just 2 months ago, we have a beautiful paper here from here, further dissect how this ETA pathway is being affected. So you should read this paper, it's really good. But the thing is that the temperature effect go beyond the ETI because it can affect the basal immunity in a genetically susceptible plant. So Bethany Yard was a former graduate student. She started this line of research many years ago now, 10 years ago. This is a genetically susceptible oilseed plant, in fact, by Pseudomonas syringae. You can see that if you, so these plants actually grown at 23 degrees the whole time for 4 weeks. Only during the 3-day infection, you know, into the comparable chamber. So it's simulating almost a heat wave. It's not experience the heat all the time. You can see that at a high temperature, you see more dramatic disease, as you can see here. And this can be replicated in Arabidopsis. It's the same. You can see more dramatic. So there's no algae involved, just basal immunity. So something is being impacted in this genetically susceptible plants. She did measurement of these defense hormones to see which hormone is being impacted. So salicylic acid, ABA, and jasminic acid. What you can see here, these are reporter genes for different pathways. For SA pathway, we usually use called PR1 gene. You can see that during normal temperature, DC3000 infection of Arabidopsis will have dramatic increase of PR1 because the SA pathway is being activated. But if you do the same experiment using 30 degree or 28 degree or 25, actually, you start to see downregulation of PR1. This is not the case for JA pathway or ABA pathway. They're actually being upregulated. So it's a selective suppression of SA pathway during this interactions. So that's the conclusion she had. She said SA defense is temperature vulnerable module of the plant immune system. And then I think this is actually quite important because SA is not only important for basal defense, but also for even, the effective chilled immunity against a variety of different pathogens. Very significant concern for future agriculture. And I'm really glad people here are really working on this intensively. And heat wave suppression of SA pathway is common. So not just in the crucifer that I just showed you. We did a tomato, I think the tomato here and rice. They, of course, grow at different temperatures. So you have to raise temperature above their normal range. And you can see suppression of SA responses. So it's probably a quite universal phenomenon. You may ask why. I don't have an answer. So just keep that question to yourself. There must be a reason. I think it's also you have to think about natural ecosystem versus the agricultural system. So there must be a role in the natural ecosystem, but it's probably not relevant in agriculture. Anyway, so we think now at least salicylic acid is one of the weak points at the elevated temperature. We should worry about this. So we went out further to see what genes in this network, which is extensive for SA. Many people have devoted a lot of time onto this, understanding how many genes are involved in SA production during pathogen infection. So you can see lots of these genes. They all impact SA accumulation, either by production or removal, okay? So it's extensive, 20 or so genes. So this was work by Danwei, Castrovolti, and Jung-Hoo Kim. They also have their own lab right now, so they all moved on. And many failed experiments. So what we try to do is to identify so-called rate limiting step. Okay, so we know that all of them are downregulated by a temperature, but if we could, can we block it essentially? So if it's a positive regulator, we want to constitutively express it to bypass the native promoter to see that can stop the SA downregulation. For a negative regulator, we do mutant. And I can tell you, none of them work. This was a 5-year journey, very hard work. And for instance, I'll give you an example, ICS1, which actually is gene that encodes an enzyme that make SA. So that was the one that we focused on a lot. We can overexpress this one and to make SA in normal temperature. Before I say this, so IS1 is downregulated. As you see here, normally it's upregulated during infection, but at 28 degrees, you practically see nothing, okay? And so what they did, this is in collaboration with Mary Wilmers at Berkeley, UC Berkeley. We basically using 35S promoter to constitutively express ICS1, so it bypass the temperature suppression in the native promoter. You can see really the assay now, usually in the wild-type plants, you need a pathogen infection to induce it. But in the ICS1 constitutive expression plants, it's always producing assay. That makes sense? Because it's an enzyme that make it, okay? And then the test was to put this, the plants look awful. So it's a growth different trade-off, I don't want to mean that, but it's small, okay? It's a very classic phenotype. But if you put on the 28 degree, it doesn't prevent suppression of assay. This is an enzyme that actually make assay. It's still not sufficient to break it, okay? So they went through all these genes, I think every single one of them, and no success until they come to the CBP60G and later on SOD1. And these are two paralogs, kind of function redundant. CBP60G is a calmodulin binding protein 60G, which means it's regulated by calcium, so it's a gray area. And this is a unique phenotype because when they did this one, they found some interesting phenotype and also some phenomenon that we're pursuing right now. So if you look at the promoter region of CBP60G, so usually you will see RNA polymerase, mediator, and a bunch of regulators. I don't want to come into this. It's very complex, the promoter. It's not clearly delineated right now. But also need another protein recently shown by Mickens lab called GBP03, which is also in human and immunity. In plants, actually, it form, I will show you, condensate. But you can see that the CBP6G gene itself is normally induced during infection, but at 28 degrees, it's not. Okay, I just told you that before. So you can actually track the transcription of CBP60G using GBPL3 GLP fusion because that delineate the activity of the promoter. So now you can do real-time imaging. So that's what we're doing, try to follow what's happening with the transcription activity at this key master regulator of assay response. And one of the phenotype of the elevated temperature is to reduce the number of the condensate formation. So you can see here that I think this is the 23 degree without or with assay, and this is a 28 degrees. So you don't see much increase here. So this is associated with the activity of CBP60G. You can also do this called a chip experiment, chromatin immunoprecipitation using different components. We did all of them, quite laborious, to see which protein goes to the promoter at different temperature. Okay, so I just give you one example. GPO3 chip experiment, we are asking whether this protein goes to the promoter at different temperature or not. You can just see this blue here. I'm sorry, here was. So this is a control promoter, so it should be negative. This is CBP60G promoter. What you can see here is this is 23 BTH, so it comes up, it binding to the promoter, but if it's 28 degree, it's not. So interestingly, not all these temperature sensitive. So this is a part that is temperature sensitive. The most chronic one in assay response is actually not temperature sensitive. So including MPL1, TGA, and some of the worky things, they are independent of condensate formation. So knowing this step is temperature vulnerable, what we want to do is for that particular work was to bypass this. Can we rescue immunity by putting the CBP6G and the constitutive promoter like 35 as promoter like we did with ICS1 to see whether that can rescue the immune response. So in this case, it does. So Columbia, as I showed you before, 23, this is 28. You see more disease here. And in this transgenic plants, there was no disease in either case. They were pretty happy. That was after 5 years. And so it was a tremendous experiment. We have now taken this to crop plants. We kind of, in the lab, we call it CBP6G technology. The first crop we were working on is the rapeseed. So club root disease is very severe right now for unclear reason. And these are root infection by a parasite, essentially, okay? You can see the club root at normal temperature and club root at warm temperature. In the field, it's pretty massive, okay? If you do disease index, you can see warm temperature actually increase infection. We have put Arabidopsis CBP60G in rapeseed. We're also doing a rapeseed CBP60G right now, but the results not in yet. You can see here that at both temperature, the independent transgenic lines has much fewer disease compared to the control. But also you can see that the increase is not that dramatic, especially with the high expression transgenic plants. This is in tomato. This is done by a group independent of us. Okay, we actually didn't know until recently they were doing this. In this case, they were using Cas9 system to basically combine heat inducible transcription to CBP60G. So it's not normally produced, but if it's a heat wave, it will produce CBP60G. It's really interesting system. I kind of cropped out many of the results. It's very detailed, but this is disease symptom here. DC3 infection with tomato have increased infection with heat here. But if you do the same thing with CBP60G expression, you see no change. Okay, so that was pretty impressive. And they also did with SOD1, which is the paralog of CBP60G, is same result. So both CBP60G and SOD1 can counter temperature-induced susceptibility. So take-home message of this part, we have identified CBP60G transcription as one of the targets for elevated temperature during basal immunity. And I think CBP60G has it's a promising target to think about to prevent assay downregulation when temperature is high. You all know this, you're doing this right now. So I think for students at postdoc, now you're thinking about what project to invest. I think this is quite important, really, in scientifically and also in practical sense. There's lots of abiotic stress need to work on. So I think this is a really exciting area. So the part of story that I think I pointed out all the names. I want to repeat it, but I want to point out collaborators: Kedong, Peizhou, Xuqing Chang. Okay, they are involved in the AVIE wall soaking projects as a collaborator. Mac Mikan and Tao Chen was involved in the heat experiment. I think with that, that's it. I think.
Yes.
Yeah. I don't want to get into the south, but actually this actually has interesting. Anyway, okay. All right. Any questions for me?